Early in hematopoiesis, a multipotent stem cell differentiates along one of the two pathways, giving rise to either a common lymphoid progenitor cell or a common myeloid progenitor cell. The type and amount of growth factor in the microenvironment of a particular stem cell or progenitor cell control its differentiation. During the development of lymphoid or myeloid lineage, stem cell differentiate into progenitor cell, which have lost the capacity for self-renewal and committed to a particular lineage. Common lymphoid progenitor cell give rise to the B progenitor cell, T progenitor cell and natural killer cell and some dendritic cell. Myeloid stem cell generate progenitor of RBC, erythrocyte, many of various white blood cell, leukocyte, neutrophil, eosinophil, basophil, monocyte, mast cell and dendrite cell and platelets. Progenitor commitment depend on the acquisition of responsiveness, neutrophil, eosinophil, basophil, monocyte and dendritic cell. Progenitor commitment depend on the acquisition of responsiveness to particular growth factor and cytokines. When the appropriate factors and cytokine are present, progenitor cell proliferate and differentiate into the corresponding cell type, either a mature erythrocyte or particular type of leukocyte or platelet generating cells, the megakaryocyte. Red and white blood cell pass into bone marrow channel from which they enter the circulation. Lymphocytes constitute 20 to 40% of body's white blood cell and 99% of the cell present in the lymph. There are approximately 10 to the power 11 lymphocytes in human body, range depending on the body size and age. These lymphocytes continually circulate in the blood and lymph and are capable of migrating into the tissue space and lymphoid organ, thereby integrating the immune system to a high degree. The lymphocyte can be broadly subdivided into three populations: B cells, T cells and natural killer cell on the basis of function and cell membrane components. Natural killer cells are large granular lymphocytes that do not express the set of surface marker typical B or T cell. Resting B and T lymphocytes are small, motile, non-phagocytic cell which cannot be distinguished morphologically. B and T lymphocytes that have not interacted with antigen referred to as naive or unprimed are resting cells in the G0 phase. Known as small lymphocyte, these cells are only about 6 micron in diameter. Their cytoplasm form a barely discernible rim around the nucleus. Small lymphocytes have densely packed chromatin, few mitochondria and a poorly developed endoplasmic reticulum and Golgi apparatus. The naive lymphocytes is generally thought to have a short lifespan. Interaction of small lymphocyte with antigen in the presence of certain cytokines induce this cell to enter the cell cycle by progressing from G0 phase into G1, S, G2 and M. As they progress through the cell cycle, lymphocyte enlarge into 15 micron diameter blast cells called lymphoblasts. These cells have a higher cytoplasm nuclear ratio and more or generally complexity than small lymphocytes. Lymphoblasts proliferate and eventually differentiate into effector cell and memory cell. Effector cell function in various ways to eliminate antigen. These cells have short life spans generally ranging from few days to a few weeks. Plasma cells, the antibody secreting effector cell of B cell lineage, have a characteristic cytoplasm that contain abundant endoplasmic reticulum to support their higher rate of protein synthesis, arranged in concentric layer and also many Golgi vesicles. The effector cell of the T cell lineage, including the cytokine secreting T helper cell and the T cytotoxic lymphocyte. Some of progeny of B and T lymphoblast differentiate into memory cell. The persistence of this population of cells is responsible for lifelong immunity to many pathogens. Memory cells look like small lymphocytes but can be distinguished from naive cell by presence or absence of certain cell membrane molecules. Fate of antigen activated small lymphocyte. A small resting lymphocyte, naive or unprimed, reside in the G0 phase of cell cycle. At this stage, B and T lymphocyte cannot be distinguished morphologically. After antigen activated, B or T cell enter the cell cycle and enlarge into lymphoblast, which undergoes several rounds of cell division and eventually generate effector cell and memory cells. Electron micrograph of a small lymphocyte showing condensed chromatin indicative of a resting cell and enlarged lymphoblast showing decondensed chromatin and a plasma cell showing abundant endoplasmic reticulum arranged in concentric circle and prominent nucleus that has been pushed to the characteristically eccentric position. Different lineage or maturational stage of lymphocytes can be distinguished by their expression of membrane molecule recognized by particular monoclonal antibodies. All of the monoclonal antibodies that react with a particular membrane molecule are grouped together as a cluster of differentiation. Each new monoclonal antibody that recognizes a leukocyte membrane molecule is analyzed for whether it falls within a recognized CD designation. If it does not, it is given a new CD designation reflecting a new membrane molecule. Although the CD nomenclature was originally developed for the membrane molecule of human leukocyte, B lymphocytes derive its later designation from its site of maturation. In the bursa of Fabricius in birds, the name turned out for bone marrow is its major site of maturation. In a number of mammalian species, including humans, mature B cell are definitively distinct from other lymphocytes by their synthesis and display of immunoglobulin antibody molecule, which serve as a receptor for antigen. Each of approximately 1.5 into 10 to the power 5 molecule of antibody on the membrane of single B cell has an identical binding site for antigen. Among the other molecule expressed on the membrane of B cells are as following. B220 is a frequently used as a marker for B cell and their precursors. However, unlike antibody, it is not expressed uniquely by B lineage cell. Class 2 MHC molecule permit the B cell to function as an antigen presenting cell. CR1 and CR2 are a receptor for certain complement product. FC gamma R2 is a receptor for IgG type of antibody. B71 and B72 are molecules that interact with CD28, important regulatory molecule on the surface of different type of T cell including Th cell. CD40 is a molecule that interact with CD40 ligand on the surface of helper T cell. In most cases, this interaction is critical for the survival of antigen stimulated B cells and for their development into antigen secreting plasma cell or memory B cell. Interaction between antigen membrane bound antibody on a mature naive B cell as well as interaction with T cell and macrophages selectively induce the activation and differentiation of B cell clones of corresponding specificity. In this process, B cell divide rapidly and differentiate over 4 to 5 days period, generating a population of plasma cell and memory cell. Plasma cell, which have lower level of membrane bound antibody than B cell, synthesize and secrete antibody. All clonal progeny from a given B cell secrete antibody molecules with the same antigen binding specificity. Plasma cells are terminally differentiated cells and many die in 1 or 2 weeks. T lymphocytes. T lymphocytes derive their name from their site of maturation in the thymus. Like B lymphocyte, they cell have membrane receptor for antigen. Although the antigen binding T cell receptor is structurally distinct from immunoglobulin, it does share some common structural feature with the immunoglobulin molecule, most notably in the structure of its antigen binding site. Unlike the membrane bound antibody on the B cell, though the T cell receptor does not recognize free antigen. Instead, the T cell receptor recognizes only antigen that is bound to particular classes of self molecule. Most T cell recognize antigen only when it is bound to a self molecule encoded by genes within the mature histocompatibility complex MHC. The difference between the humoral and cell-mediated branches of the immune system is that the B cell is capable of binding soluble antigen, where T cell is restricted to binding antigen display on self cell. To be recognized by most T cells, this antigen must be displayed together with MHC molecule on the surface of antigen presenting cell or on virus infected cell, cancer cell and grafts. The T cell system has developed to eliminate this altered self cell to the normal functioning of the body. Like B cell, T cell express distinctive membrane molecules. All T cell subpopulation express the T cell receptor, a complex of polypeptide that includes CD3. And most can be distinguished by the presence of one or the other two membrane molecule CD4 and CD8. In addition, most mature T cell express the following membrane molecule CD28 and CD45. CD28, a receptor for the co-stimulatory B7 family of molecules present on B cell and other antigen presenting cell. CD45, a single transduction molecule. T cell that express the membrane glycoprotein molecule CD4 are restricted to recognize antigen bound to class 2 MHC. Where T cell expressing CD8, a dimeric membrane glycoprotein, are restricted to recognition of antigen bound class 1 MHC molecules. Thus, the expression of CD4 versus CD8 also define two major functional subpopulation of T lymphocyte. CD4 plus T cell generally function as T helper cell and are class 2 restricted. CD8 plus T cell generally function as cytotoxic T cell and are class 1 restricted. Thus, the ratio of T helper cell and T cytotoxic cell are 2:1 normally, but it may be significantly altered by immunodeficiency disease, autoimmune disease and other diseases. Th1 response produce a cytokine profile that support inflammation and active mainly certain T cell and macrophages, whereas T helper cell 2 responses activate mainly B cell and immune responses that depend upon antibody. Another subpopulation of T lymphocyte called T suppressor cells has been postulated. It is clear that some T cells help to suppress the humoral and the cell mediated branches of immune system. But the actual isolation and cloning of TS cell is a matter of controversy and dispute among immunologists. For this reason, it is uncertain whether TS cell do indeed constitute a separate functional subpopulation of T cells. Some immunologists believe that the suppression mediated by T cell observed in some system is simply the consequence of activities of TH or TC subpopulation whose end result in suppressive. Thank you.